Good day, everyone. These videos will discuss about the lipids, the structure of lipids, the properties of lipids, and the biological significance of lipids to organisms. I am Mr. Ong from College Tingkatan 6 Seliputra. Previous lessons, we talked about the carbohydrates. So today, we would like to discuss about the lipids. So we will describe the structure, properties, and the distribution of triglyceride, which is known as fat, phospholipids, and steroids, and state the function, okay, the biological function of triglyceride, phospholipid, and steroids. And we also differentiate between the saturated and unsaturated fatty acid. Lipids are a heterogeneous group of compounds that are categorized by the fact that they are soluble in non-polar solvent, such as ether and chloroform, and are relatively insoluble in water. So we call it as a heterogeneous group because they consist of different types of compounds. Therefore, the lipids are not macromolecules and they are not polymer. So they have no monomers to make up this lipid. Okay. So they are not macromolecules, okay, or they are not the polymers. So therefore they have no monomer. So lipid molecules have hydrophobic properties because they consist mainly of carbon and hydrogen with few oxygens containing functional groups. Some lipids are used for energy storage. Others serve as structural components of cell membrane, and some are as important hormones. Amongst the biologically important groups of lipids are fat, or we call it triglyceride, phospholipids, carotenoids, steroids, and waxes. In these videos, we only discuss fat, phospholipids, and steroids. What is fat? Fat is also known as triglyceride. It's the most abundant lipids found in organisms. And triglyceride also is known as triacylglycerol. And commonly, we call it as fat. Triglycerides are compact, insoluble in water, and can be stored at high concentration without requiring water as a solvent. The triglyceride is a form of reserve energy storage because when metabolized, they use more than twice as much energy per gram as do carbohydrates such as glucose. Carbohydrate and proteins can be transformed by enzyme into fat and to be stored within the cells of adipose tissue, or we call it as adipocytes of animals, especially for those animals living in cold climate. Triglyceride also found in and stored in some seeds, okay, and fruit in the forms of oils, which is the liquid form. So other important role, okay, of the glyceride in living things, okay, other than as a storage energy, okay. In aquatic animals, the fat can act for buoyancy. For hibernating animals, they store food as additional fat layers below the skin. That means in the adipose tissue. And also act as insulator, okay, heat insulator against the loss of heat, or we say reduce the heat loss. And around the visceral organs for protection, such as the fat layers they found in the to cover, okay, to cover or to around the heart, okay, for protections to absorb shock. Triglyceride also release twice as much water as do carbohydrate when oxidized in cellular respiration, And then the water they produce is known as metabolic water. So this is important for the development of very young birds and reptiles while they are enclosed in their eggshells and then without obtaining waters from the external source. So, and the daily metabolisms of animals, okay, in the habitats where the water is scarce, okay, such as camel and kangaroo rat. So, for this type of animals, they will oxidize the fat they store, okay, by cellular respirations to produce the metabolic water. A triglyceride molecule consists of one glycerol. Okay, this is a one glycerol. Okay, joined to three fatty acids. Okay, by ester bonds. These are the ester bonds. Okay, via condensation. Glycerol is a 3-carbon alcohol that contains 3 hydroxyl groups, and then a fatty acid is a long, and usually it's a 16 or 18 carbon atoms, okay, or we call the hydrocarbon chain. The either is so they are unbranched, they are unbranched hydrocarbon chain. So and then they have a functional group, which is the carboxyl group. So this carboxyl group will react with the hydroxyl group of the glycerol, so to form an ester bond and release one water molecules. So since they have three what we call the condensation reactions taken place, so 3 water molecules are formed. Okay, as you see the reactions here, you can see a triglyceride molecule is formed by a series of 3 condensation reactions. So remember, it's 3 condensation reactions, and don't forget to write 3 water molecules are produced. Okay, in each condensation, the equivalent of a water molecule is removed, okay, or is produced as one of the glycerol hydroxyl group react with the carboxyl group of the fatty acid by condensation. Okay, remember the process, and resulting in the formation of a covalent bond known as ester bond. So if you break down the triglyceride, so the process the process is known as hydrolysis. So by addition of 3 water molecules, and with the enzyme lipase. Okay, so this will occur in during digestions. The triglyceride is hydrolyzed to produce 1 glycerol and 3 fatty acid. Okay, so by the enzyme lipase. Okay, saturated and unsaturated fatty acid. So from the triglyceride, okay, this is the hydrocarbon chain, or we call it fatty acid. So the fatty acids can be classified, okay, according to the presence of double bond, okay, between the carbon atoms. About 30 different fatty acids are commonly found, okay, in lipid. So normally they have an even number, so 16, 18, 20, 22, okay. So for example, oleic acid with 18 carbons is the most widely distributed fatty acid in nature and is found in most animal plant fats. So they have what we call the double bond, okay, 1 double bond here, okay. And the fatty acids can be classified, okay, as a saturated and the unsaturated fatty acid. So like just I mentioned, those depends on the double bonds. Okay, saturated fatty acid contain the maximum possible number of hydrogen atoms, okay, on their carbons. For example, palmitic acid and stearic acid. Palmitic acid is 16 carbons fatty acid and is commonly saturated fatty acid. So you can look at this, the fatty acids change. So this is the functional group. So carbon 1, 2, 3, 4, and so on until carbon 16. Okay. So all the carbons, they're joined by single bond, single covalent bond. And in the stearic acid, okay, stearic acid also is a saturated fatty acid which has the chemical formula of 18 carbons. Okay, hydrogens, 36 hydrogens, and 2 oxygen. So the carbon 1 is here, carbon 2 is here, and so on. In contrast, unsaturated fatty acids include one or more adjacent pairs of carbon atoms joined by a double bond. Okay, they say one adjacent pairs of carbon atom joined by double bond or more. Okay, so for example, look at this one. There is what we call a double bonds presence. So that means these carbons they are not fully saturated with hydrogens. So the other example here is here they have 2 double bonds. Okay, then the carbon number we count from not from the functional group, but it's from the nearest to the double bond. Okay, so here's carbon 1, carbon 2, carbon 3, and so on. Okay, the unsaturated fatty acids with 1 double bonds in the hydrocarbon chain, we call it as monounsaturated fatty acid. Then those with more than 1 double bonds, we call them as polyunsaturated fatty acid. So this is known as monounsaturated fatty acid. This is polyunsaturated fatty acid. For example, oleic acid is a monounsaturated fatty acid. Okay, the oleic acid, which is a monounsaturated fatty acid, like just now we have mentioned. And the linoleic acid, okay, linoleic acid, they are polyunsaturated fatty acid, okay, with 2 double bonds, whereas the alpha linoleic acid, okay, contain 3 double bonds. Okay, this one commonly we call it as ALA. You can get it from the infant's milk powder, so you can see ALA. Okay, and then it is one of the omega-3. Okay, this is omega-6, this one is omega-9. Okay, so omega-3, that means the fatty acids contain 3 double bonds, whereas the omega-6, they have 2 double bonds, and the omega-9 with 1 double bond. So why we call them as the omega-3, omega-6, and omega-9? This is because the omega-9, that means the double bonds is found on the carbon 9. Okay, carbon 1, 2, 3, 4, 5, 6, 7, 8, 9. So double bonds is on the carbon 9. So we call it as omega-9. So these are omega-6, that means the double bond, the first double bonds is found on the carbon 6. And then omega-3, that means the first double bond is found on the carbon 3. When we have understand about the saturated fatty acid and the unsaturated fatty acids, now we can go further to the saturated fats and unsaturated fats. So they are different. For the saturated fat, okay, saturated fat, you can see the saturated fat actually they consist or made up of 3 saturated fatty acid. Okay, all the fatty acids they found in the triglyceride molecule are saturated fatty acid. So these fat molecules is known as saturated fat. Okay, so such as animal fat and a solid vegetable shortening. So that tends to be solid at room temperature as usual. Okay, that you learn in SPM. Okay, so this is because the fat molecules they arrange compactly. When they arrange compactly, so they will develop the van der Waals interactions between the fat molecule. Okay, even though the van der Waals interactions they are weak interactions, but when they have many, so they are strong when they accumulate. So due to the long hydrocarbon chain. Okay, and then these van der Waals interactions that tends to make this fat, okay, or the saturated fat becomes solid at room temperature. In contrast, the unsaturated fat that contain at least one unsaturated fatty acid. Okay, so when they contain at least one unsaturated fatty acid, so when they arrange, the triglyceride molecules they arrange together, so they have more spacings, so they do not align closely. So these spacings, they will limit the van der Waals interactions between the triglyceride molecule or the unsaturated fat. So these unsaturated fats that tends to be liquid in room temperature. Okay, so when we have learned complete the triglyceride molecules, now we move to the phospholipid. Phospholipid is another lipid that consists of one glycerol molecules that attach, okay, to two fatty acid, and the other hydroxyl group will attach or binds to a phosphate group. Okay, like show you here. Okay, so this one here we call it as a hydrophilic head, and the tail, because consists of fatty acid, so we call it as hydrophobic tail, which is non-polar. So normally we show the structure of the phospholipid like this, okay, with a sphere here and two hydrocarbon tail. Okay, the hydrophobic tail, which is the hydrocarbon chain fatty acid. And then the hydrophilic head here consists of the glycerol molecule and also the phosphate group. If the phosphate group here, okay, this is a glycerol molecule, this is the fatty acid chain. If the phosphate group, which is the negatively charged, they bound with a choline molecule that containing nitrogens, okay, this is called a choline molecule, which is positively charged because of the nitrogens. So this phospholipid molecule is known as lecithin. Okay, lecithin. Easier for you. So when we say the lecithin, if the question asks you to describe the lecithin, don't forget to describe the choline molecule that binds to the phosphate group of the phospholipid molecule. So this is the example of lecithin, or we call it as a phosphatidylcholine. Okay, phospholipids are amphipathic lipids. Okay, when you see amphipathic, so that means they got two different polar. Okay, different pole. So for example, here, like just now we have mentioned, here, this is the hydrophilic head, and here is the hydrophobic tails. So the two ends of the phospholipid differ both physically and chemically. Oh. The portions composed of glycerol, phosphate, and the choline, so we call it as hydrophilic and readily water soluble. So whereas the hydrophobic tails, they are not soluble in water. And then you can see here for the lecithin, they have a kink. The kink is due to the double bond, okay, or we call the unsaturated fatty acid, and due to the cis double bond, okay. So this kink is important. Okay, it's important for the in the plasma membrane, because the kinks, they prevent the closely alignment or the closely arrangement of the phospholipid in the plasma membrane, so that they can provide the fluidity, okay, or the flexibility of the to the plasma membrane. Okay, and then the amphipathic properties, okay, of the phospholipid cause them to form the phospholipid bilayer, as you learn in SPM. So you see this phospholipid, the hydrophilic head, which is attracted to the water molecule, and then the hydrophobic tail, they away from the water molecules. Okay, so they will form the cell membrane or the plasma membrane as the barriers, okay, to isolate, okay, the cell content with the surrounding. Then the phospholipid also act as surfactants, as a surfactant in our lungs, okay, to lower the surface tension. Because on the inner surface of the alveolus, they have a water film. Okay, this water film or the water layer allow the respiratory gases such as oxygen, okay, to dissolve, okay, and also allow the carbon dioxide, okay, from the cell to dissolve into the water before they diffuse. Okay, so if just the water layer, so the water layers will make they will have the high surface tension. Okay, they have a high surface tension as you learn in the water topic. Okay, then this high surface tension they will make the what we call the alveolus, okay, to stick together. Okay, and then difficult to be inflate, okay, during inhalations. So the surfactant, this surfactant is produced, okay, by one of the alveolar cells, okay, we call the type 2 alveolar cells. So they will produce the surfactant. So these surfactants will lower the surface tension of the water molecules, of the water lah. So that they prevents the lungs from collapsing, okay? So that the alveolus can be easily infected, okay, during inhalations. And then the phospholipids are also as a source of acetylcholine when they're hydrolyzed. So the choline group can be reused as the acetylcholine, the neurotransmitter, okay, neurotransmitter in the nervous system. And also in the myelinated neurons, the myelin sheath actually consists of phospholipid as well. Okay, so the phospholipid also play the important role as the component of myelin sheath. And the phospholipid also involved in the transport of fat between gut and liver in mammalian digestions. So from here, they form the micelles, okay, the phospholipid, they form the micelle, and then they contain or carry, okay, or transport the cholesterol and the triglyceride as well. So like this one, they have what we call HDL, okay, high density lipoprotein, okay, and low density lipoprotein and very low density of lipoprotein. Okay, so among these lipoproteins, so the HDL is the good one, so which can helps to eliminate or carry away the LDL, that means we call the bad cholesterol, okay, from the artery. And then the HDL is the good cholesterol, okay. All right, then we move to the final part in the lipid, which is steroids. A steroid consists of 4 attached carbon rings. Okay, these 4 attached carbon rings and side chain that extended from this ring. Okay, so 3 of the rings contain 6 carbon atoms. 3 of the rings that contain 6 carbon atoms, and the 4th ones that contain 5 carbon atoms only. Okay, so the length and the structure of the side chain that extended from this ring distinguish one steroid from another. So that means all the steroid, they will have these 4 attached carbon rings. Then the different one is the side chain. Okay, what determines the types of the steroid. Okay. The steroid can occur or found in both plants and animals. The biological importance of steroids are cholesterol, bile salt, vitamin D, sex hormones, and other hormones secreted by adrenal cortex. So now we look at the cholesterol structure. Okay, so they have 4 attached rings, okay, carbon rings, and then the side chain. Okay, here is a very long hydrocarbon chain, and here with a hydroxyl group. So that means here this is the polar group here, okay, and then the other, all of these they are non-polar, or we call the hydrophobic. So this is hydrophilic here. okay, but it's a very small. And the rest can see the testosterone, estrogen, and the progesterone. They are the sex hormones. And this is cortisol, the hormones that are produced by the adrenal cortex. So all of these hormones, they have the 4 attached carbon rings as the structure, as the steroid skeleton. And then the difference on the side chain. Okay, the side chains will be different. So that determines the type of the steroid. So cholesterol, okay, because our syllabus are more focused on the cholesterol. So we look at the cholesterol structure, and cholesterol is synthesized in the liver, and the formula is C27H46 and O. Okay, they have the 27 carbon. So the 4 attached carbon ring and the carbon side chain are hydrophobic ends, okay, which is lipid soluble. Okay, so the side chain with the hydroxyl group is hydrophilic end, which is the water soluble and can interact with water molecules. So therefore, in cell membrane, okay, the cell membrane contains cholesterol. Then you can see the cholesterol here. So the arrangement and the position, okay, the hydroxyl group is near to the hydrophilic head, and the rest, the hydrophobic components, okay, or the hydrophobic ends, they will found between in the phospholipid, no, in the what we call the fatty acid chain, okay, the hydrophobic tails region. Okay, so this is the position, okay, or the location of the cholesterol in the plasma membrane. But when the excess cholesterol in blood forms plaques, okay, they form the plaques on the artery wall, so the risk of cardiovascular diseases increases. Okay, so they will cause the atherosclerosis, for example. So we will discuss these cardiovascular diseases in the next semester. And the cholesterol, they are the precursor, okay? Cholesterol molecules are the precursors to make bile salt, okay, sex hormones, and the hormones of the adrenal cortex. So even though we know the excess cholesterol, they can harm or they will increase the risk for people to have cardiovascular diseases, but the cholesterol also is a good component, okay, and that is an essential component for organisms because the cholesterol is the precursor. Okay, precursor means the raw material is used to synthesize the bile salt, sex hormones, and the hormones from the adrenal cortex. So that means all of this, they will derive from the cholesterol. Okay, so like vitamin D, okay, vitamin D, they also is a steroid closely related to the cholesterol. Okay, so the ultraviolet radiation in sunlight, they will convert this provitamin D to vitamin D. So you see the provitamins and the precursor here also have the steroid skeleton for attached carbon rings. Okay, so in summary, we have learned about the structure of the triglyceride molecule, which is the fat molecules. Okay, and then how is the fat molecules or the triglyceride is formed? So through a condensation. Okay, and then by one glycerol with the 3 fatty acid. Then these 3 fatty acids are joined with the glycerol by the ester bond, okay, through the condensation. And then 3 water molecules are produced. And then we also learn the structure of the saturated fatty acid and the structure of unsaturated fatty acid. And then from the saturated and the unsaturated fatty acid, we also learn how is the and distinguish the difference between saturated fat and unsaturated fat. And also we learn the phospholipid structure, the phospholipid functions, okay, and also the examples of the phospholipid, which is a lecithin, and with the choline positively charged group that binds to the phosphate group. And we also learn the cholesterol, okay, the steroid example, cholesterol and others hormones that derive from the cholesterol, and we also learn some of the function of the steroid. Okay, so from here you can see the triglyceride, the phospholipids, and the steroid. Okay, even though they are lipids, but they have different structure. Okay, and then they also have different function. So, but once they have the common is all of the lipids molecules, okay, all of these triglyceride, phospholipid, and the steroid components, okay, steroid compounds. So they are insoluble in water, okay, but they are soluble in organic solvent. Then the other examples are waxes. So I just show you the diagram, okay, where are the waxes can found. So in the feather, okay, and also in the fruit, the skin of the fruit, and also the beehive. Okay, they contain the waxes. Okay, and so this is what we have learned. Okay, and for the next lessons will be protein. So that's all for today, and thank you very much. Bye.